Everyone, for this week's lab we're going to be studying the control of heart function using the hearts of frogs, the spotted leopard frog, Ranapipians. For this lab, there's a number of aspects of preparation for the lab. One of those is that we are going to be working with some substances that are toxic. So they're in these small dropper bottles right here, like so. So substances such as epinephrine. And so for that, you're going to want to come with safety goggles, just to be safe. Along with that, we'll also have gloves as another form of protection. And for this lab, we are going to be sacrificing a set of frogs. We have to work with a freshly prepared specimen that still has a functioning heart. And because frogs are ectotherms, their hearts actually can continue functioning for quite a while after the frog has been sacrificed or euthanized. So they're really a fantastic system for learning a lot more about how heart function is regulated. So this does involve the sacrifice of some animals, and because of that, you need to come to lab prepared to put the animal tissues to the best possible use. The reason that we do this is there's not really a good alternative to actually studying these processes using a living tissue. So we haven't made the decision to use frogs lightly. And you should also be aware that there are detailed protocols in place to ensure humane treatment of the frogs, as with any animal that we keep in captivity, up to the point where the frog is euthanized. We've had to make some revisions to our protocols in the last 2 years, so I'm going to be using some new anesthesia protocols with the frogs. I don't know what the impacts are going to wind up being on heart function, so this is partially going to wind up being a larger experiment to document the effects of that anesthesia on this heart protocol. But I feel that especially for those of you that are planning to pursue some career in medicine, that it's critically important that you get direct hands-on experience working with this type of tissue and working with these physiological preparations. Just because I wouldn't want you to go in and start performing surgery on a person without having any prior experience working with different types of biological tissues. So I know that this can be a little bit challenging for some individuals. And you'll be working in groups and working together through this process. So I just want you to come, emotionally prepared and focused to put our time in lab to the best possible use. So having said all of that, the preparation for this lab is also a little more involved than some of our previous setups have been with LabChart. So the next thing that I'm going to do in this video is walk you through some of the details of that preparation process so that hopefully it can go as smoothly as possible. And the first thing I want to point out is we're going to be measuring the strength of heart contractions using our friend, the force transducer. So let's go over and take a look. Okay, so here's our force transducer. And in this case, the force transducer is now connected to my ring stand using a fine scale adjuster, because for this preparation we're going to want to have a very good direct connection with the frog's heart, especially because the frogs we work with, their hearts are fairly small. They will produce a relatively weak force, so we need to have everything lined up well to pick up a good signal. So what this unit does is it has a little knob on the back that will let you adjust the height of the entire unit. And then it also has a screw knob down at the base that will let you just very gradually raise and lower the height of the unit to get a very precise height. This particular one's a little finicky, but it will give you about a centimeter of space to work with. Alright, so the next part of this setup process is going to involve getting the power lab configured. So with the force transducers, Just as a reminder, the first thing you're going to want to do is to turn on the Power Lab unit. You're going to wait for the green light to turn on and then plug in your force transducer. Okay, so force transducer is plugged in there to channel 1. Once you have that plugged in and ready to go, then launch lab chart. And this time through, you'll notice something a little bit different with the force transducer. So we'll just open up a new file. We'll just work with channel 1 for right now. And what I want you to notice is I've got a little hook here running through the blade on the force transducer. So what we're going to do now is we're going to calibrate channel 1 using a set of little weights. So here's a little 10-gram weight, and I've just tied a piece of string around that weight, and then on the other end I've got a big loop going. So there's my loop. And then also a little 2-gram weight, and I've got a little loop for my 2-gram weight as well. And these calibration weights are found in a cute little box that is in the red toolbox that's on your table. And you'll also see, if you open up that toolbox, there should actually be a list of tools. We'll get into that in a minute. So to calibrate your force transducer so that it's recording in mass units instead of in voltage units, first thing we're going to do is we're going to go up here and we're going to switch this down to 50 millivolts. So it's a more sensitive range for this instrument. Then I'm going to start lab chart, and you can just see the trace running through there. And I'll start out with my 2-gram weight. What I'm going to do is I'm going to try to avoid a lot of swinging when I do this, but I'm just going to hook my 2-gram weight onto the force transducer. You can kind of see where I've wiggled this around. And then once I get a relatively steady reading, I'm just going to lift this back off of the force transducer. Okay? And then in this trace, this point here where I have the lowest negative voltage, so negative 1.634, I can see that up in this upper right corner, or I could also pull out this channel 1 measurement, right, negative 1.55. Negative 1.68 appears to be my low spot there. I'm just going to write that down somewhere in my notes as the voltage that corresponds to 2 grams. And then I'm going to repeat this process now with a 10-gram weight. And you'll notice when I put the weight on, I'm going to have a positive voltage recording. So you can sometimes calibrate off of the positive voltage recording, but you'll notice this is pretty wiggly. So usually I have better success calibrating off of the negative voltage recording when I remove that weight from the transducer. So this has started to stabilize out. So now I'm just going to lift this weight back off. And I'm going to take this negative voltage reading at the valley here. You can also do something like over in DataPad here, instead of the mean, you can just record the minimum value. So negative 11.9062. So that corresponds to my 10-gram mass. And now what I can do is if I go into the channel 1 drop-down menu and I go to units conversion, so 0.1, I'm just going to take the absolute value of those measurements. 0.1 was then 1.6844 millivolts. And that corresponded to 2 grams, or milligrams. And then the 11.9062 corresponded to 10 grams. Okay? Then if I click OK, now you'll notice that my x-axis is rescaled into grams. The paper clip here is going to add mass, but that's going to be a constant mass throughout the subsequent recordings. And so now I can measure force production in grams. So the next thing I want to walk you through is a little tour of the tools in your toolbox, which you'll be using for the preparation of your frog heart. And I'm also going to note, I am not going to demonstrate this with a frog, because when we decide to work with vertebrate animals, whether it's for education or for research, one of the principles we use is minimization, so using the minimal number of animals necessary to accomplish our goals. So I would consider using a frog for this purpose to be an excess use of an animal. And instead what I'm going to do is I'm going to provide you with a link to a video from another class as the class has walked through this frog heart preparation process. And that video will show you the process of preparing the frog's heart. Instead, what I'm going to show you at the moment are just the tools that are in your toolbox that are going to wind up being useful. So again, I'll be preparing the frog for you, so you'll be receiving a freshly euthanized frog. There can sometimes be a little bit of remaining activity in the frog, but I'll be double pithing the frogs, so that should reduce activity overall. Once you have your frog, you're going to want to lay your frog in a dissecting tray with its ventral surface up, and then you're going to use some scissors to cut through the skin. And your toolbox should have 2 sets of scissors, so it's going to have a set of larger blunt tip scissors and a set of smaller fine tip scissors. Either set of scissors should be just fine for that initial purpose. But you will also hit a point where you reach the frog's rib cage, and for that it's often easier to remove bone using a set of bone shears. I've got 2 sets of bone shears, so when the time comes I can come around and help you cut through some of the bone to lift up the frog's rib cage and help expose the heart. Then once you've cut through the skin, the other things that can be very helpful to use are your toolbox should have a set of these jawed forceps. So if you look closely, you know it's a little fuzzy, but there are little jaws on the tips of these forceps, and those jaws are really helpful for holding skin without slipping. You will also have some blunt-tipped forceps, which you may also use. And then along with all that, another tool that can be really useful is one of these blunted probes to move things around. And you'll see that there are a couple of other tools in your toolbox as well. So your goal is going to be to expose the frog's heart and open up the pericardial sac. So for opening up the pericardial sac, by that point it's usually useful to hold the pericardial sac with the jawed forceps and then use the fine scissors to cut it away so that the heart is exposed. And your goal with doing that is going to be to expose the tip or the apex of the heart. So that's where the heart's single frog heart's ventricle is located. And with that, then what you want to do is you're going to have a little tray on your table that says "small pieces do not lose." That includes the little hook for the force transducer, and the other thing that's in that is a very tiny little heart clip. And with this heart clip, you're then going to take a piece of string. And you'll see there's a tiny little loop on the heart clip. And so you want to thread some string through that loop. Tie it off. And then this is a little spring-loaded clip with a tiny little set of jaws. So you can, if you squeeze together, you'll open up those jaws, and you want to secure those jaws around the apex of the heart. And then take the other end of your string, and you're going to attach the other end to the hook on the force transducer. And so then once you get everything dialed in just the right location, the beats of the heart will be detected and quantified by your force transducer. All right, now I'm going to show you a set of measurements that you can take from a heart trace. So here's an example trace that was recorded during a previous semester by another group of physiologists. And one thing I want you to notice with this trace is they've used comments to note when they've applied different substances to their heart. So atropine, acetylcholine, I think there's epinephrine in here somewhere. Yep, epinephrine. So with all of that, there are 3 different measurements that we can take from these data. So I'm just going to select a section of the data here, open up DataPad, and the first thing we can do is just get a measurement of heart rate. So that again is cyclic measurements, average cyclic rate. We're in channel 2 in this case, so I'm just going to click OK, and now I've got my average cyclic rate of about 2.4 beats per minute. Along with that, if we want to get a measure of the contractile strength, the contractile strength is equal to the overall change in height here, so from the maximum to the minimum. So again, we are in cyclic measurements, and that's just going to give us, or be calculated as the average cyclic height. Okay, so that's our contractile strength. And then the third parameter that's of interest to us is a measurement of overall heart muscle tone. And for that, that is simply just calculating the mean of our data. So tone will be, again, in units of grams. Okay, and with those 3 measurements, we'll be able to characterize heart function under any particular condition. So that's it for this video. For getting ready for lab, you definitely want to take some time to read through the lab handout in advance, so you have a game plan in place. Typically things work best if you have one person or team working on getting the force transducer set up and getting lab charts set up, and have another person or team focus on preparing the frog. And then once the frog is ready and your force transducer is calibrated, then you'll be in a good position to start collecting data. And definitely take some time to read through and examine the chart that walks you through the sequence for applying different treatments to your heart. And after reading through the introduction, you should also be able to put down your predictions for what's going to happen to your heart's rate and to its amplitude under the different treatments that you're about to apply. Then it'll be time to do science and find out if your actual results match your predicted results.